All right, good afternoon. Thank you all for coming to the colloquium this afternoon. I am really delighted to introduce Rees Garmann. Rees is our newest addition to the faculty in chemistry and biochemistry. And even better, Rees really spans the two kind of traditional divisions of chemistry and biochemistry, because he's a physical chemist studying viruses. And so this is great. I stand to learn a lot from this seminar. Rees got his bachelor's degree at Berkeley and his PhD at UCLA. And then, like a lot of Californians, chose to try out the colder weather, and he went to Harvard to work with Vinathan Manoharan, who's one of the pioneers in really looking at the physics of viruses. And Rees did some outstanding work. In fact, a video on the New York Times website has been showing some of the work that he's been on, examining the kinetics of virus self-assembly. And then we were lucky enough to nab him, and he's been with us for all of about 6 weeks at this point. And I'm really looking forward to hearing about his research on self-assembly. Rees, I'm going to hand it over to you.
Thanks very much, Andy. Can everyone hear me? Yeah, okay. Well, I'm very excited to get to talk to everyone. And I'd like to encourage everyone to interrupt me at any point in this talk. I'd much rather get some questions from people than finish everything. So I'll be talking for the next little bit here about two of my favorite subjects. That's self-assembly and RNA viruses. And I'd like to just start by kind of giving you some definition or what I mean when I say self-assembly, and also what I mean by RNA viruses for some people, some computational people who may not know what those are, even though given the pandemic, I think we're all more familiar with RNA viruses than ever before. Okay, so self-assembly, what I mean when I say self-assembly, is just any process in which an initially disordered system of particles becomes ordered spontaneously without any external intervention. Okay, and so water freezing is a perfectly good example of self-assembly. And you can find videos of water freezing, examples of it on YouTube all over the place. I think this is a particularly charming video. This is a bottle of water that somebody had left in their car at -9 degrees Celsius. And it came back in the morning. I'll play it again. And it was liquid. Gave it a crack with a flashlight. And you can see that an ordered phase, an ice phase, is forming within this supercooled water. It's supercooled because the water is below its freezing point. And so if you had a microscope sensitive enough, what you would be seeing within this system is something like what's shown here, right? You have liquid water. It's all disordered, jumbling, tumbling around, jiggling due to thermal energy. And spontaneously, if the temperature is dropped down low enough, so I'm going to try and move my...
Excuse me.
Each box, yes.
You say no external intervention, but here the temperature comes from outside.
Ah, right. So I consider the temperature to just be a part of the system. That's just. Okay. Is that clear?
Yeah.
Okay. So then, but there is a key difference in that the water bottle had to be whacked with something, and I'll describe what's going on with that process a bit later in the talk. But so here the disordered water molecules have interactions between them. The interactions are weak interactions, hydrogen bonding interactions between the water molecules. And when the temperature is above the freezing point, those interactions are sufficiently weak that they never have time to take hold. But when the temperature is dropped down, and the thermal energy in the system becomes comparable or less than the interactions between the water molecules, then an ordered phase emerges in which all of the molecules, because of these hydrogen bonding interactions, line up, form a crystal lattice, and take on a very, very ordered structure. And this ordered structure at temperatures below the freezing point are a minimum, is a minimum of the system free energy. So this new structured system forms at equilibrium by a thermodynamically favored process. It is the favored thermodynamic phase. Okay, so that's as good of an example of self-assembly as I can think of. But it's interesting that this process wasn't originally called self-assembly. It was originally called crystallization. Most people know this process known as crystallization. And that's because the term self-assembly wasn't coined until the 1960s. And it was first used to describe not the formation of crystals, but instead the formation of these little creatures here, RNA viruses. So these are simple RNA viruses. They're made entirely of a protein shell made up of order 100 proteins, all the same protein, repeated many times to form a more or less spherical shell, and a genome molecule that's packaged inside of the shell. So the protein shell protects the genome, and the genome can be made of nucleic acid, either DNA or RNA, but in the case of RNA viruses, the genome's made of RNA. And the simplest viruses, the simplest RNA viruses, use RNA that's written as plus sense single-stranded RNA. And what that means is that the sequence of the RNA can be directly translated into proteins. And why these are the simplest viruses becomes clear as soon as you look at what the viral life cycle is for these viruses. So the way that these viruses propagate is that the viral RNA has to get out of the protein shell and into a host cell. And because the RNA is written in the positive sense as messenger RNA, it can be immediately translated by the host cell ribosomes. And those ribosomes have to make at least 2 proteins in general. One of those proteins is what's called the replicase protein. And the replicase is a protein complex that takes the original viral RNA that came into the cell and makes many, many copies of it. And the second protein that the virus has to make is what's called a coat protein. And this is the same protein that makes the shell. So the viral RNA codes for this coat protein. Many copies are made by host ribosomes, and the newly synthesized RNA strands and the newly synthesized coat protein molecules come together and form new virus particles. And these particles then leave the cell and go on to infect new cells. Okay, and here's a picture of the process happening. It's a picture of an E. coli cell being infected by my favorite virus, which is called bacteriophage MS2. Okay, and what you're seeing here, can everybody see my mouse? Andy, can you see my mouse here?
No.
How about now? Now you can see it? Okay, cool.
Yes.
So what you're seeing here is an outline of an E. coli cell. This is 15 minutes after it was infected by an MS2 virus. And it's in very bad shape. You can tell because the cell is bursting. Here's a bit of the cytoplasm that's starting to leak out of the cell. And in fact, you can even notice that in the upper corner up here, that there are freshly made virus particles that have been replicated to such high numbers that they're actually crystallizing inside of the cell. They're forming a little ordered patch. So based on the size of this little patch of viruses, you can estimate that there are something like 1,000 viruses in this cell. And so the MS2 was able to replicate itself something like 1,000 times in the course of 15 minutes in one cell. And these 1,000 viruses can then go on to infect 1,000 new cells. And you can see very quickly how virus replication and propagation takes off. Okay, but where we get interested in this process as a self-assembly process is we can take the virus particles from this infected E. coli cell and we can purify them away from all the cellular debris. Get a very, very pure sample of this wild type MS2. And you can see on the left here that MS2 is a nice spherical particle. It's about 28 nanometers in diameter. And it appears to be very, very regular in these electron micrographs. And indeed, you can take these virus particles and you can crystallize them and you can do X-ray diffraction studies on the protein capsids that make up the virus. And you can measure to basically perfect precision the position of every atom in every protein in this viral shell. And that's what's shown on the right here. This is a crystal structure of the virus capsid. And it's made up of precisely 178 copies of the coat protein and 1 copy of a slightly different protein called A protein, which won't be important for the purposes of this talk. So, yes?
Please, sorry,
yeah.
2 questions. One is, can you go on the prior slide where you had the massive burst size that MS2 does? And
Yeah.
so in the prior step, do you have in vivo any compartments in the formation of MS2? It does procapsid and then it packs the DNA. Does it form the procapsid and lays it in some parts of the cell and then packs the DNA, or it does it everywhere?
Right, so that's a good question, and that's the main part of the talk is what is the mechanism by which these virus particles form. But in RNA viruses, different than DNA viruses, you don't first form an empty shell, which then gets packed full of the genome. Instead, the assembly of the capsid occurs at the same time as the packaging of the RNA. It's a co-assembly process where the proteins organize themselves around the genome molecule. And there is no compartmentalization in this process. At least I'll say there's no compartmentalization in this process. And in particular because bacterial cells don't have any cellular compartments. They have no membrane enclosed compartments. So unlike eukaryotic viruses, eukaryotic RNA viruses, which form so-called virus factories, which are invaginations of internal membrane structures. The RNA phages don't do that because there's no internal membranes to deform.
Mm-hmm.
Does that make sense?
Yep.
Okay,
Thank
cool.
you.
So we're at a very advanced stage of structural virology in that we can know the order of viral capsids to essentially perfect resolution, right? And that's shown here on the right. And these structures are, there's no other way to put it, they're beautiful. They have very high symmetry. So this is a rotating view of the MS2 capsid. When I say symmetry, they have icostahedral symmetry, which is the highest point group symmetry. And by that, I mean that they have a 5-fold axis of symmetry, a 3-fold axis and a 2-fold axis, which I hope you can see here. And this is the same symmetry and the same organization that's shared with, for instance, a soccer ball. So in a soccer ball, you have 12 black pentamers and 20 white hexamers, at least the old school soccer balls that I grew up with playing with. I know that the new soccer balls look nothing like this. And if soccer balls don't help you visualize what a virus looks like, it's the same structure as a geodesic dome. This is a Buckminster Fuller's geodesic dome. And you can see again that in the geodesic dome, you have an alternating pattern of pentamers, 5 triangles, alternating with patches of 6 triangles as hexamers shown in blue. Okay, and in the case of the virus, that means that there are clusters of 5 proteins and 6 proteins, and they alternate in a very precise way in order to form a closed shell. Okay, but unlike the geodesic domes that we see on the right that are put together by construction workers, by people, these viral capsids form spontaneously. And what I mean by that is you can take purified solution of the virus coprotein and a second purified sample of the virus RNA, totally purified, simple RNA molecules in buffer solution, simple protein in buffer solution. And you can mix them together, and without doing anything, you go have a cup of coffee, you come back, and in your test tube are virus particles that structurally are indistinguishable from those that are born in a natural infection. Okay, so this assembly process really is an example of self-assembly. There's no special ingredients that need to be present other than the coat proteins and the RNA in order for these structures to form. It's not something special about the biology of the host cell. ATP hydrolysis, for instance, an input of energy is not required. This process happens spontaneously at room temperature, driven just by thermal energy and interactions between the proteins and the RNA. And so the main question, one of the main questions of this talk is, how does this process occur? And we can get kind of a picture in our minds of what it might look like by looking at computer simulations. These are coarse-grained Brownian dynamic simulations in which the proteins are represented by these pentagon structures. And there are certain beads on the pentagon shown in yellow that interact with the genome molecule that's shown in red. So there's an attractive interaction between the pentagons and the genome. And then there's also a lateral interaction that's programmed into this simulation between the pentagons, between these green patches on the pentagons. And so these simulations show you can resolve very precisely where each protein goes and how intact structures might form. And there are some key takeaways that we can learn from these simulations. And the key takeaways are, one, is that self-assembly is a random process. So all of these motions are due to random kicks of the solvent, and there is no directed motion. Things do not know where to go at any point in time. They're simply just jiggling around, forming a closed shell. The RNA plays a key role. I'll play this movie again so you can see it again. So you can see that the RNA is playing a key role. It's interacting with proteins kind of throughout this whole process. The interactions between all of the components in the system are weak. Necessarily, they have to be weak for reasons I'll discuss in a bit. But you can tell that they're weak because proteins will come and bind and unbind and rebind and this type of cascade. And the assembly timescales, because these interactions are weak, are necessarily very broad. So what I mean by that is that individual binding events between proteins or between proteins and the RNA occur very fast, bind and unbind on very short timescales, but it takes a relatively long time for this giant capsid assembly to form. And then we learn we get some clues as to what maybe the pathways for the self-assembly process are from simulation. And the takeaway for me is that the pathways are anything but simple. There are multiple pathways that are observed in the simulation. So for example, in the top here, you see an example of an ordered pathway in which a small cluster of proteins forms at one end of the RNA. And that small cluster then grows into a complete shell surrounding the RNA. That happens under certain conditions, meaning certain values for the interaction strengths between the proteins and the RNA. But if you change those interaction strengths, then you can get different assembly mechanisms. So on the bottom here, I'm showing a mechanism in which first the proteins glom onto the RNA in a disordered way. And then later they rearrange all bound to the RNA as one big complex into this final ordered structure. And you can switch between these pathways and others by changing the input parameters of the simulation. And another key takeaway is that oftentimes the simulation doesn't end up in the right structure. So many times if you have the interaction slightly off, you form terrible misformed things, monster structures. And even when the interactions are right, you still get some fraction of particles that don't quite assemble right. Okay, so as an experimentalist, I'm not satisfied to just watch simulations of capsids assembling. I would like to take a movie of a virus capsid assembling and see what these real systems do with real interactions between proteins and real interactions between the RNA. Okay, so we need a dynamic measurement. Up until this point, all of the measurements I've talked about, all the experiments I've talked about on viruses are all static measurements. X-ray crystallography is a static, gives you a static picture of a virus capsid once it's already formed. It does so in very high resolution, but you get no dynamic information. So we need a dynamic technique. And one of the best dynamic techniques for watching processes in time resolving assembly processes is static light scattering. And so the idea in static light scattering is you have a light source and you shine some illumination beam onto a sample of assembling virus particles that are assembling within a cuvette. And then you measure the amount of light that is scattered from the assembling particles with a detector. Okay, and if you orient your detector at 90 degrees to the incident light source, then you have very nice separation between that incident beam. You have a dark field technique, and you can detect relatively sensitively increases in scattering that occur as virus particles grow. As the particles grow, they scatter more light, and you can detect that increase in scattering in your detector. And these experiments have been done for 20, 30 years at this point. And what you get is some type of curve, these curves that start at low intensity, and then as the virus assembles, you see an increase in intensity and some plateau as the viruses take on their mature structure. But this technique is averaged over all of the virus particles in the cuvette. And so it's very hard to work out from this averaged measurement what the individual self-assembly pathways look like. In other words, what is happening at the scale of a single virus. And the reason that it's difficult is because these viruses might be assembling each individual particle, might be assembling out of synchrony from each other. And so what you're measuring is an average of a bunch of virus particles that are out of synchrony with each other, and it's very hard to reconstruct what's going on at the single virus scale from these average intensity curves. Are there any questions at this point? I should be careful to ask more.
So, Rees, how complex are these capsid proteins compared to, I don't know what kind of scale you use for proteins, but are they remarkably simple or not?
Well, relative to other proteins, they look normal. They're globular proteins. They're 30, 40 kilodaltons, normal size. But I mean, relative to the coarse grain simulation, they're much more complex. Yeah.
I have a question, may I ask? Is there any binding or attraction between the capsid proteins themselves, or I guess what makes the capsid more likely to stay together once it's put together if the bindings come undone sometimes?
Right. So yes, there is definitely there's an attraction between capsid proteins. In real viruses, it's mainly due to hydrophobic interactions between hydrophobic residues that line the protein-protein interface. But those interactions are weak, and they're weak yet strong enough such that once a capsid forms, it's a stable structure. It is the thermodynamically favored state.
Thank you.
Any other questions? Okay, cool. So the question we asked was, can we do a light scattering measurement, but not on a bulk ensemble of assembling viruses, but can we do it on a single virus? Can we watch a single virus assemble itself in real time? And there's a bunch of reasons why this is a very hard thing to do. First and foremost is that viruses are very small, so individually they scatter very little light. And to get a feel for how little light they scatter, you can compare what's called the scattering cross-section of a virus, essentially the size, the area of a shadow that's cast by an individual virus particle. You compare that to the smallest area over which you can confine a laser beam, which is the diffraction limited spot size. And you can infer from that at best, you can, a virus will scatter 1 out of every 10 to the 7 photons that impinges upon it. Okay, so at best, the contrast that you can get from a single virus is something like 10 to the minus 7. And the situation gets, yep.
So, Rees, one question. So you say that it's 10 to the minus 2 nanometers squared, but a protein on the virus would be already on the order of 3 nanometers, 5 nanometers. So how the virus could have such a low scattering factor?
Yeah, it's a good question. I think it comes down to the fact that the proteins are like essentially translucent. You know, they're not opaque. So their scattering cross-section is not their physical cross-section.
Got it.
So the situation is much, much worse for the components of the virus. So the scaling with which the scattering intensity depends on mass or on size is that it goes as the mass of a particle squared. This is true for sub-wavelength particles, particles that are smaller than the wavelength of light. And what that means is that if you have 1/10 of a virus capsid, it scatters 1/100th the amount of a full virus capsid. So detecting early intermediates in the assembly pathway is very, very hard. Okay, the second main challenge is that viruses diffuse very quickly. So even if you manage to get a virus within the diffraction limited spot of your laser beam, it's going to leave very fast. And the third problem is that the time scale for assembly is very long, right? So you have to, well, it's broad, right? You have to have fast time resolution in order to capture individual binding events. And you have to have very long time resolution in order to watch the entire assembly process take place. And that's a big problem because that big dynamic range rules out techniques like fluorescence microscopy. So you might think, well, I'll just label each protein with a fluorophore, and then I can watch as the fluorescence signal builds up on individual particles. But you can't do that because fluorophores bleach. And they bleach after they emit so many photons. And so you can't get a broad dynamic range out of individual photons. You can't take fast measurements for a long time. You can either take measurements spaced out for a long time, or you can take measurements that are taken in rapid succession, but you can't get both. Okay, so the way that we solved these problems is to use a technique called interferometric scattering microscopy. And this technique was developed by Philip Kakura in England, and also by Vahid Sandagar. So this technique is slightly different than the direct light scattering technique, and I'll describe very briefly how it works. So here you take an incident laser beam, and excuse me, I gotta move this thing again. And you pass your laser beam through a sample which contains virus particles. And some portion of the light gets scattered from the virus, as shown here, same as in the previous setup. Okay, but another thing happens when you shine this laser beam onto your sample. And that is that a fraction of the beam gets reflected from the glass-water interface. So now you collect that reflected light and the scattered light back down through the microscope objective and image it onto a camera. Okay, and the key here is that now you're not simply imaging the scattered wave from the virus particle, but you're imaging the interference between the scattered wave and the reference wave. And if you get your setup in the correct geometry, you can get constructive interference between the scattered wave and the reflected wave. And that interference ends up boosting the scattering signal, such that you can detect very faint scatterers, even single viruses. And to give you a feel for kind of the size of this increase, you go from having a contrast of 10 to the minus 7, like we were talking about, now down to something like 10 to the minus 2. So 5 orders of magnitude increase in contrast. Okay, and the proof of this is that you can take MS2 virus particles, very puny viruses, some of the puniest viruses on the planet that we know of, and they're 3.6 megadaltons. And you can add them to a microscope cover slip, image them with the interferometric scattering microscope, and you can see individual MS2 particles. These particles are coming from solution and then binding to the cover slip, and you only see them once they're bound. Okay, but what we want to see is we want to see the assembly of particles. We don't just want to see them coming down from solution, we want to see them growing. And so the way that we do that is we tether the RNA strands to a glass cover slip, and then we flow in the coat proteins in a disassembled state. And we image the coat proteins as they assemble around the RNA and form capsids, form virus particles. So this is and we've done controls to make sure that indeed virus particles, virus capsids can assemble around surface tethered RNA strands. Okay, so this is an example of what the movies look like. Here is, you're watching, you don't see the RNA molecules because we've subtracted their intensity away in the background. We do a background subtraction. But you see, as we flow in coat proteins, we see dark spots emerging. Here the video is looping again. And these dark spots correspond to individual virus particles that are becoming darker or more intense as a function of time as more proteins bind to the RNA. So is this clear what everyone's seeing here? Each one of these individual dots is a self-assembling, self-assembling coat proteins assembling around an individual molecule of MS2 RNA that's tethered to a glass cover slip. Okay, so this is the dynamic information that we were after. Okay, so here you can measure the intensity of each individual particle as a function of time. And what I'm showing here is one of these intensity traces for this particular particle. You see that it starts off at low intensity, and then after some amount of time, it jumps up to a higher intensity and then plateaus. And it's important to point out that in this technique, we resolve no structural information. So this is in some sense the polar opposite of the structural virology techniques that I showed at the beginning of the talk. There you get perfect spatial resolution. In structural virology techniques like X-ray crystallography, you get perfect structural spatial information about the arrangement of the proteins in the capsid, but you get no dynamic information. Here we get dynamic information, time-resolved information about the size of the particle as a function of time, but we lose all of the structural information because we use light and optical wavelengths of light, which are larger than the capsid itself. So we lose all information because of the diffraction limit. But nonetheless, we get a very sensitive measure of the number of proteins that are bound to each RNA molecule. And so the intensity shown here is directly proportional to the number of coat proteins in this assembling particle as a function of time. And we can calibrate our microscope using intact virus particles. So we pre-calibrate our microscope so that we know exactly where full capsids should be, what intensity they should be. And indeed, we see that this particular capsid grew and then plateaued to a size that is consistent with a full capsid. So we think that this particular event is representative, this trajectory, this trace of intensity is representative of a viral capsid forming.
Rees, this is Elio.
Hey.
Can I ask you the most naive question conceivable?
Yes.
How about altering the temperature when you do this? Going to right above the freezing point. Does that help you?
So that's a great question. We haven't played with temperature. This is actually, Andy, you remember this? You asked this question during the last talk that I gave. It's a very precise question. So it's a
kinetics
great
question.
If Andy asked it. And the answer, I'm afraid to say, is just it's technically difficult to change the temperature in this experiment because the microscope is very sensitive to thermal drift. So we would have to, the simplest way to do it I can think of would be to change the temperature of the entire room. But it's a bit of an undertaking, and we haven't done that yet, but I'd love to see some temperature dependent data. Okay, but at room temperature, under the conditions that we've been doing, we get measurements for many individual capsids in the same experiment. Okay, and so here's the second particle, right? And it does more or less the same behavior, only it starts at a different time. Okay, and so on for the other virus particles that are forming in this experiment. Some of them, interestingly, don't grow to the correct size. So this one in red grows to a slightly bigger size. And if I have time, I can discuss why that is. I'm getting concerned that I won't have time to do that, though. Okay, so the question is, we have all of these individual assembly measurements. What can we do with them? And the first observation that we make, right, is that there is a very broad distribution in the time at which individual particles start growing. Okay, some start growing as early as 100 seconds into the experiment, but some don't grow all the way out until 300, 400, even 500, depending on the day, depending on the experiment. They can go out thousands of seconds before they start assembling. Okay, so what's going on with this broad spread in the start times? And this goes back to the example with the freezing water. So in this example, you have water. It's liquid water, but it's at -9 degrees Celsius. And yet, so the water would prefer to be ice, right? Its thermodynamically favored state is ice. And yet you have water that's sitting there super cooled. And the reason that that is stable or is allowed to happen is because in order to transition from liquid to solid, you need to overcome an energy barrier, a free energy barrier. There's a barrier to forming the new phase. And that barrier is supplied by the guy who whacks the bottle. Okay, so by whacking the bottle, he creates some vibration or some bubble or something on which an ice crystal can nucleate and then grow throughout the sample. Okay, and the physics of this nucleation process or of this energy barrier has been worked out long ago. It's called classical nucleation theory. And it has to do with the idea that in order to form a new phase, the new thermodynamic phase being the ice, the crystal phase, you get an energy benefit, a free energy benefit for forming the new phase that scales as the volume of the new phase that you've created. But in forming this new volume, you've also created now a surface, an interface between the ice and the water phase. And you pay a penalty for forming that surface. And the penalty that you pay is proportional to the amount of surface you have, which goes as the size of the new cluster, new patch of ice squared, right? Okay, so early on in the formation, as you start to form a little ice cluster, the surface term dominates. And so the energy penalty is larger than the energy benefit that you get for forming this new phase. And it's not until the ice cluster grows beyond a critical size, shown here, that you actually end up becoming, it becomes favorable to form the new ice phase. And so that is why there is a so-called nucleation barrier, because a cluster has to grow above a certain size before it becomes stable to grow. And that's what the person had to supply when he hit the bottle. Okay, so the same appears to be true for virus capsids, is that in order to form a shell, enough proteins have to get onto the RNA and form a large enough cluster that it becomes stable to grow. Same type of argument as for the ice crystals. And this isn't a new idea. So nucleation and growth has been applied to virus capsids before. But this, now we have a way of measuring this nucleation barrier. And so can we use the classical nucleation models to understand what's going on in the assembly process? And so the first thing we can do is we can make a measurement of how many virus particles start growing as a function of time. So that's basically how many of these traces cross this red line as a function of time after we've added the coprotein. And this is a cumulative distribution of those times shown on the right. Okay, and the cumulative distribution is fit very nicely by a single exponential. And that's really a smoking gun to suggest that this nucleation process is occurring in a single step. So the nucleation models predict that single step nucleation should have a distribution of start times that follows this single exponential. Okay, so we think we have evidence that the nucleation step is a single step nucleation step. Furthermore, we can ask, what is the size of the critical nucleus that needs to form before the growth of the capsid shell becomes favorable? And it turns out that we can't directly measure the size, but we can infer something about the largest cluster that, or we can put a maximum onto the size of these subcritical clusters. And the way we do that is that we measure the fluctuations in the intensity before the assembly takes off. And so classical nucleation theory tells us that before the virus shell grows, you have proteins binding and unbinding, then clusters forming and disassembling as they try to overcome this barrier. And so we would expect to see fluctuations on the order of the size of the subcritical clusters. And in fact, the size of the fluctuations that we see prior to nucleation are consistent with just the noise in our measurement, which tells us that any subcritical clusters that are forming are smaller than the noise in our instrument, which is, we can calibrate, is smaller than about 6 dimers. So we know that the critical nucleus size is about 6 dimers or less. Okay, but that just opens up a whole new can of worms, because a small critical nucleus the size of 6 dimers is tiny compared to the full capsid shell. And so if the nucleus really is this small, 6 dimers, how does the rest of the capsid know how to grow and form its complete structure? And I thought I would have had time to go into some of our thoughts as to why or how the mechanisms by which the viral capsid grows in such a regular way. But I'll just point out another feature of our data, since I have another minute, is that once the nucleus forms, it appears that the proteins readily bind to the RNA and form a capsid in this monotonic growth. In other words, there's no backtracking steps. You don't see capsids growing and then disassembling and then having to reform and grow before they close. Essentially, once they start growing, they zip right up into the correct structure. And so something interesting is going on that allows them to do that. And there's a bunch of theories in the literature currently as to why that is. And it's up to future experiments to try and rule them out. So I'll just leave you with a teaser, which is that one of the theories is that the RNA, something about the structure of the viral RNA, is helping to guide the coproteins get into their correct final state. But evidence for that, direct evidence for that hypothesis is lacking. So I'll end there and then take any questions if anyone had any. Oh, I should zip to the end and just mention all the people that helped with this research. So this is the team that I've been working with, Ofer and Tim and Aaron and Vinny at Harvard, and then Fernando and Daniel have joined my group at SDSU, and we're starting to get started in the lab. So we hope that we'll have some answers to some of the questions that I left hanging soon. Thank you very much.
All right, thank you, Rees. That was really outstanding. So I have a couple of questions while I let other people formulate theirs. So totally unfair question, but it's computational sciences colloquium. So do you have ideas for what contributions computer simulations of this or other kinds of modeling might make to this field?
Yeah, certainly. So the answer is at present, computation has to be involved. And the reason is because we're trying to infer a dynamic structural process, a process that occurs with dynamic structural rearrangements. And we're doing so with a technique that is fundamentally blind to structure. So the measurements that we make cannot tell us what's going on at the level of protein configurations. And in order to infer that type of stuff, we need computer simulations.
So then, I'm sorry. So then the question becomes, I mean, I've done computational work where there's like, you know, there's one data point that we're trying to match, right? And you match it and you go, well, you know, that looks good. But so how many, you know, degrees of comparison are there such that you would really trust a model beyond, you know, getting the right kind of 2-parameter curve that you were showing?
Right. So I guess I would say that the burden is on us at the moment. We need to perform these measurements as a function of all the interesting variables that Elio and you have brought up. So we need to do these assembly measurements, generate kinetic data as a function of temperature, as a function of protein concentration, as a function of ionic conditions, because we have these great experimental knobs. By changing the salt in the system, we can change the electrostatic interactions, which we know dominate the protein RNA interactions. So we can effectively tune the strength of protein RNA interactions by changing salt. We can change the protein-protein interactions by changing pH, because residues that line the protein interfaces titrate at different pH values. And so we have, in principle, a very nice way, and because we're working in vitro, because we're working with purified components, we can do very precise experiments in which we vary all of these variables and measure these assembly traces, and then hand over these big data sets to computational modelers with the hope that with enough of these parameters varied, we'll have enough data points to help constrain your models.
Oh, that's great. I'm sorry, one last question. Could you say a little bit about the RNA protein interactions? Because one thing I was curious about is once, you know, in the simulation you showed, you know, the RNA reaches out and gloms onto the protein. Can the protein then slide around to facilitate formation of the structure, or is it kind of locked in? Are these phosphate interactions, or what part of the RNA is interacting with the protein?
Yeah, these are great questions, and these are questions that to some extent have been very well studied. So back in the 70s, some of their very first protein RNA interaction measurements done by Ilke Uhlenbeck at Colorado, These were done precisely between MS2 coat protein and different RNA sequences. So the thermodynamics of that interaction, the kinetics have been largely worked out. And it turns out to be a combination of general electrostatic interactions between the negatively charged phosphate groups on the RNA and positively charged residues that line the interior of the coat protein. A combination of those, but then also very highly specific, sequence specific, dependent on the RNA sequence interactions between certain bases and pockets in the coat protein fold. And these interactions lead to a very specific tight binding that is actually used throughout molecular biology as a tool set. So people use MS2 coat protein and MS2 RNA as a way to get to measure protein in live cells. They take a fluorescently labeled MS2 coat protein and they attach to some RNA of interest one of these so-called operator hairpins. And you can now get a picture of where this operator containing RNA is within a cell based on the fluorescence from the MS2 coat proteins. So all the information is in principle, I think, there and available, or a lot of it is.
Oh, that's great. Thanks. I'm sorry. So let me throw it open.
Could you get rid of the slide so we can see people?
Oh, yeah, sorry.
So Rees, I'm trying to, this is Peter Salomon. I'm trying to, here, maybe understand the self-assembly sort of beyond the getting started. It seems like you have just one long ramp, so it doesn't, but the water molecules assembling do keep attaching, retaching, attaching, retaching, and I would expect more of that in the self-assembly you're looking at. Why is it so processive, if I may?
Yeah, right. So I would just point out that what we're measuring is an average number of proteins that are bound. So there still could be rapid fluctuations that happen on timescales faster than we can observe. So maybe that's part of, or I'm sure that some of that is going on and we're just not sensitive to it. But the average number of proteins appears to increase monotonically. And so that is an open mystery. I don't know how that's happening, because the proteins have to add in precise positions.
Well, as long as they keep trying, and then when the real position happens, it's stable enough that it doesn't come apart, I think that part will work. I'm just
but something I left out, and I should have spent more time on it, because the capsid is constructed by pentameric and hexameric groups. The coat proteins that are binding and forming the shell have to, in some sense, make decisions as to whether or not coordinate with 5 neighbors or 6 neighbors. And so it's not a trivial process for them to assemble. It's not as if the correct configuration is always the same as the capsid grows. In fact, there are different configurations that have to be adopted in a very precise way.
It's always a pentamer edge lining up with a hexamer edge, right? So as long as you control the bonds along those edges, or what's available along those edges, I think.
How does a protein know to form a pentamer? So they don't come in as pentamers and hexamers. They come in as individual monomers, and they have to form the hexamers and pentamers on site within the growing shell.
That's not what your simulations looked like. It looked like that whole patch came in.
That's right. So that's the point, is the simulation was a coarse-grained simulation. All of the proteins that were coming in were already pre-assembled pentamers. There were no hexamers. So it's a very oversimplified system. That's why we need to do experiments.
Well, or better computational experiments.
Sure. But those are the cutting edge at the moment. So.
Could I ask Oliver, you've had your hand up for a while.
Yeah, so my question is, what stops you from using a laser with a smaller wavelength if resolution is the problem?
Yeah, so that's a good question. We use essentially again, it's a practical thing. We started off using a 400 nanometer laser, and we burned through our optical components. So we actually burned holes in some of the mirrors that we were using in the microscope objective. So that was a huge expensive mistake. But so it turns out that as you go to lower and lower wavelengths, the system starts absorbing that radiation. It becomes ionizing and a problem.
Wenjung Chen.
Hi, so thanks for the talk. I just wanted to ask, so some viruses don't completely assemble the capsid while they're in the cell, like most famously HIV. So how does that affect so what effect does that have on like this kind of study?
Right, so I would say, if I'm being conservative, that what we're learning about are really about these simple RNA viruses that consist entirely of a protein shell and an RNA molecule inside. And many RNA viruses are more complex than that, including, of course, coronavirus and also HIV viruses. These viruses have, in addition to what I just described, an entire lipid envelope that is studded and decorated with transmembrane proteins in it. And so there, the assembly process is much more complex. It involves the lipid membrane. It involves forming curvature within that membrane, and oftentimes interactions between the assembling coat proteins in the membrane and the RNA. It's a whole much more complicated process. The viruses that we're studying are the simple ones, are viruses like polio virus, many plant viruses, these bacteriophages.
Does the lipid membrane like sort of provide it with the environment with which to do the assembly?
Right, so it's assembly at a surface. That's right, yeah.
Thanks.
Tony, got a question again?
You're muted, Tony.
Yeah, great question, great talk, Rees. I have two questions. One is, have you explored to disentangle the contributions of the RNA assembly experiments with shorter RNA strands to try to form quasi-empty capsids?
Right, yeah. So we have experimented with shorter RNA strands. I don't know if we've gone short enough to make an empty particle, but we've cut the RNA, for instance, by a factor of 10. You still have about 400 bases that are participating. And there the kinetics are different and the particles may be smaller, so it may actually affect the size of the capsid that forms. But we have started doing those experiments. We just don't have a complete picture that I can share with you about what the results are. But you're totally right that that's another great experimental parameter that we can vary for people like Andy and Peter, which is that we can vary not only the length of the RNA, but we can also vary the sequence of the RNA. And we can start introducing mutations into the coat protein that make it behave in predictably different ways, changing its protein RNA interactions and protein-protein interactions.
Thank you.
So when the proteins assemble into pentamers or hexamers, are the edges different?
Yeah, so this is I should be careful. I mean, so the print Tony should answer that. What am I talking about?
You're throwing that one back to me. Okay, yeah. So Peter, I have to say this is very new. So until very recently, we would have classified MS2 as a virus with T3, meaning 12 pentamers and 20 hexamers. But a year and a half ago, we published this paper showing that the organization might be more, the dimers might be organized in a way that don't need to be forming explicitly hexamers or pentamers. But that's just very new. And actually, Rees and I were talking about the possibilities on how these new ways of building the icosahedral capsids that these viruses form may be affecting the assembly differently. And as I said, this is, I would say, cutting edge. Now, that's what we're learning right now. Until very recently, during 60 years, we thought that things were formed by pentamers and hexamers, and now we start getting a few more ideas. In this case, they form dimers. I don't know, the dimers would be stable. Would you say that, Rees?
Yeah, the dimers are stable in solution. You have to do very cruel things to the protein to get the dimers to disassemble.
So the dimers, for what we have observed recently, they form kind of rhombs, and these rhombs would come together, but I don't think that anyone has assembled those rhombs on simulations or anything. So we have very little insight on how they could be forming the pathways. You saw these simulations with pentamers, they've been simulations with triangles, but not necessarily with the type of tiles that now we would expect from Rees' system.
Okay, Peter, I'm going to call time. Okay. All right. Thank you, everybody. Rees, thank you so much for a great talk. Really appreciate it. So,
Very nice talk.
hey, everyone, have a
Great talk, and have a nice weekend, everybody.
That's right. Long weekend, rest and recovery day on Monday.